Hello everyone and a very warm welcome to my channel. In this video we will be talking about ICSE biology. We will be taking up the chapter structure of chromosomes, cell division and cell cycle, and we will be revising it by means of various questions. So let us start. Differentiate between the following. The first question here is to differentiate between chromosome and chromatid. So what are chromosomes? Chromosomes are condensed and coiled chromatin fibers. And what are chromatids? These chromosomes duplicate into two sister chromatids. Okay, so क्रोमोसोम क्या होते हैं? क्रोमोसोम जो क्रोमैटिन थ्रेड साथ में जुड़ करके कंडेंस होके क्रोमोसोम बना देती हैं, और ये जो क्रोमैटिड क्या होते हैं, यही क्रोमोसोम डुप्लीकेट होते हैं, टूटते हैं या स्प्लिट अप करते हैं, तो ये क्रोमैटिड बनाते हैं. क्रोमैटिड फॉर्म मतलब क्रोमैटिड बनाते नहीं है, क्रोमैटिड जो दो सिस्टर क्रोमैटिड जुड़ी रहती हैं जो फॉर्म अ क्रोमोसोम, और यही जब सिस्टर क्रोमैटिड्स अलग अलग हो जाती हैं, अलग अलग, तो दे वी कॉल देम क्रोमैटिड. सेपरेटली दे आर क्रोमैटिड. अब नाउ वी विल ट्राई टू डिफरेंशिएट बिटवीन सेंट्रोसोम, सेंट्रियोल्स एंड सेंट्रोमियर. एनी ऑफ द थ्री कैन बी आस्क्ड और द टू कैन बी आस्क्ड इन डिफरेंशिएट बिटवीन टाइप ऑफ क्वेश्चन. तो सेंट्रोसोम क्या होता है? इट इज़ अ सिंगल कॉम्पैक्ट ऑर्गेनल प्रेजेंट इन सेल रिस्पॉन्सिबल फॉर सेल डिवीजन. Centrioles are rounded bodies radiating ester rays split up from centrosome. And what is centromere? The point of attachment of the sister chromatids. तो हम ऐसे बात करते हैं. सबसे पहले सेंट्रोमियर की, तो जब सिस्टर क्रोमैटिड्स जुड़ी होती हैं, जिस पॉइंट पे वो जुड़ी होती हैं उसको हम सेंट्रोमियर कहते हैं. सेंट्रियोल्स क्या होते हैं? जो सेंट्रोसोम ऑर्गेनल होती है, जो न्यूक्लियस के ऊपर प्रेजेंट होती है. जब सेल डिवीज़न इनिशिएट करता है, धीरे धीरे सेल साइकिल स्टार्ट हो रही होती है, तो ये सेंट्रोसोम सेंट्रियोल में स्प्लिट अप कर जाती है और दोनों साइड ऑफ दी सेल में चली जाती है. और सेंट्रोसोम कहते हैं जब सेंट्रियोल्स में नहीं टूटा होता है सेंट्रोसोम, तो एक सिंगल कॉम्पैक्ट ऑर्गेनल की तरह न्यूक्लियस के ऊपर प्रेजेंट होता है. अब एस्टर एंड स्पिंडल फाइबर्स. Ester are small radiating structures arising from centrioles. The ester rays are the rays which radiate from the centriole. And spindle fibers, these ester rays, they form the spindle fiber on which the chromosomes attach themselves. So these both are different things. In mitosis 2 daughter cells are formed, and in meiosis 4 daughter cells are formed. You can differentiate between mitosis and meiosis in various ways, and a table is given in our book. You can go through it. Here we are differentiating between the two on the basis of number of daughter cells formed. So in mitosis 2 daughter cells are formed, whereas in meiosis 4 daughter cells are formed. Okay, now we will be discussing about the odd one out type of questions, and we will be naming the category of the other three belong to, of which the other three belong. Okay, now the first question here is adenine, guanine, cytosine and thyroxine. So here what is the odd one out? It is thyroxine, because we know there are 4 nitrogenous bases on the DNA, and they are adenine, guanine, cytosine and thymine. And here what it is written? It is written thyroxine. So definitely it is wrong. What is thyroxine? Thyroxine is a hormone which is secreted in the thyroid gland, and which regulates the basal metabolic rate. These are other things. So what do the other three belong to? What is the category of the other three? The category of the other three is nitrogenous bases. Odd one out thyroxine, category nitrogenous bases. Now we will talk about the second question, G1 phase, anaphase, G2 phase and synthesis phase. Here the odd one out is anaphase, because the other three phases are the stages of interphase, and after interphase there is prophase, metaphase, and then there is anaphase. So here odd one out is anaphase, and the category to which the other three belong is stages of interphase. Now third question, telophase, interphase, luteal phase and prophase. Here the odd one out is luteal phase, and the category to which the other three belong is stages of cell cycle or the stages of M phase. Okay, so why luteal phase is an odd one out here? Because luteal phase is not associated with cell cycle. It is associated with associated with the ovaries, etc. matlab or you can say the menstrual cycle. One of the phases, luteal phase. Okay. Mitosis. Now we will talk about the fourth question, mitosis, meiosis, cell cycle, amitosis. So here cell cycle is the odd one out, because the other three are basically different types of cell division. Okay, so these are the four odd one out type of questions from this chapter. Now we will talk about arrange in logical sequence type of question. First question is metaphase, anaphase, prophase and telophase. So this is not the correct order. The correct order is prophase, metaphase, anaphase and telophase. Now you must remember this. It's very important in specific order. These are what are these? These are the stages of M phase. Now second question, G2 phase, G1 phase, synthesis phase and M phase. Now this is not the correct order. The correct order is G1 phase, then synthesis phase, G2 phase and then M phase. Okay. Now we will talk about the fourth question, DNA, nucleosome, cell and chromosomes. This is not the correct order. We will talk from inner side to the outer side, मतलब अंदर से बाहर की बात करेंगे, from outside इन, और ऐसे बोल सकते हैं. ओके, सो सबसे अंदर जो है वो डीएनए होता है. डीएनए से न्यूक्लियोसोम फॉर्म होगा, न्यूक्लियोसोम से क्रोमोसोम फॉर्म होगा और क्रोमोसोम से सेल. कहने का मतलब है सेल है, सेल के अंदर क्रोमोसोम है, क्रोमोसोम के अंदर न्यूक्लियोसोम है और न्यूक्लियोसोम के अंदर डीएनए है. तो सबसे छोटे से सबसे बड़े यूनिट की तरफ हम जा रहे हैं यहाँ पे. Now in this question, label and name the following structures. So what is this? This is a nucleotide. Nucleotide are repeating components which are present in a cell basically. So these repeating components, what are they made of? They are made of a pentose sugar. Like you can see, it's like a pentagon, 5-sided pentagon. We say na 5-sided polygon is called a pentagon. So it is a pentose sugar. It is a base, nitrogenous base, about which we talked about, adenine, guanine, cytosine and thymine. And what is this? This is a phosphate group linked here. The second one here is chromosome, not very well drawn, but still it can be used for explanation. So this is a chromosome, and the point of attachment of the sister chromatids here, centromere. Now this one is DNA. You can see there are rungs formed here, or rungs present here. These rungs are basically nitrogenous bases linked with each other. So thymine pairs with adenine with double bond, and guanine pairs with cytosine with triple bond. So DNA, what is the structure known as? It's DNA. What is the bond between thymine, and what type of bond between thymine and adenine? It's double bond. And what bond? Hydrogen bond. And bond between guanine and cytosine, it's triple bond. It's also hydrogen bond. So it might be asked what type of bond is present in between the nitrogenous bases in the DNA. So it is hydrogen bond. Okay. Now this question pertains to name the stages of mitosis and write their characteristic features and draw the diagrams. So the first stage here is interphase. In interphase, the cell, there's not much change in the cell. The cell is as it is, and it is preparing for the M phase. Okay. So you can see only while drawing these stages, you have to draw only those organelles which are associated with cell division. Okay, and you'll not draw other organelles. All the other organelles are present in the cell. Definitely there is Golgi bodies, endoplasmic reticulum, etc., but you will draw only mitochondria, etc., but you will draw only those organelles which are associated with cell division, so that your diagram is not messy. Okay, so this is the first diagram. You can see it. This is the second diagram. Early prophase. In early prophase, nucleolus starts to disappear. Centrosome, I told na that centrosome splits and the centrioles start moving apart, and these are the two main points which you have to remember. What happens in early prophase? Nucleolus starts disappearing. Nucleolus present inside the nucleus, now it's not there, so it starts to disappear. This chromatin threads, this thread-like structure, start condensing to form chromosome, and the centrosome, which was a single compact organelle here, has now split up and it's trying to move to the poles. I hope you have understood. Now the third one, it's the late prophase. In the late prophase, see, the centrioles have actually come to the either side, and they have started to form these esterases. I have forgotten to told here, the esterase formations begins here. The esterase formation, because of the esterase formation, the spindle fibers also start forming, and as a result, the chromosomes attach themselves. The chromosomes attach themselves on these spindle fibers. Okay. Here the characteristic feature is that nuclear membrane starts disappearing and spindle fiber formation starts occurring. What is this phase? Late prophase. Now this is the fourth phase here, metaphase. In metaphase, what happens, that chromosomes align themselves in a, this is a very characteristic feature, that chromosomes align themselves in an equatorial plane. They align themselves in a straight line here, and the diagram is not very clear, I'm sorry for that, but you can see the diagram in your book also. Basically I'm trying to revise all the concepts. So chromosomes, what happens in metaphase, chromosomes align themselves in a single line. So whenever there is a diagram in the question paper and chromosomes are aligned in an equatorial plane like this, you at once you should think that, oh, this is metaphase. Okay. And while reading, you should remember the stage prior to the one stage and stage after one stage, because such type of questions are very frequent and very frequently asked. Now, early anaphase, what happens in early anaphase? These sister chromatids, which we talked about earlier, they start to move apart. They split up and they start to move apart. Where they start to move? They start moving near to the centriole. Here they start moving here. Now in late anaphase, what happens, that these sister chromatids who have started to move to the either sides actually come to the either sides of the either sides near to the centrioles. Okay. Now what starts happening? Now again we are coming from where we started. A furrow starts to form in early telophase. Even the furrow formation starts in late anaphase also, but in early telophase the furrow formation is very very prominent, and the furrow deepens, and basically two cells are being prepared. So what happens? A nuclear membrane starts appearing, a nucleolus starts appearing, and these and we have seen that these chromatids have moved apart. Now these chromatids start forming the chromatin threads are starting to appear. Okay, and nuclear membrane starts appearing. Now in the late telophase, we have actually the cell cycle has actually been completed, and the cell has now been divided into two daughter cells with full grown, with all the organelles. Now what all organelles it has? All the organelles which are present, and the plus which we are talking about here, that the nucleus is there, the nucleolus is there, chromatin threads are there, centrosome is there, and two daughter cells are formed. Okay, so now what these daughter cells will do? These daughter cells again, they'll go in the process of interphase, G1 phase, G2 phase, G1 phase, S phase, G2 phase, then after that M phase. So again these cells will prepare themselves for the cell cycle, and this cycle of cell cycle will continue ever and ever. Okay, and that is how we are living. Okay, now we'll talk about the name the following type of question. The first question is coiled and condensed chromatin fibers. So these are chromosomes. Second one, repeating component of which single DNA is composed. So repeating components, whenever you see repeating components, nucleotide should be the only thing revolving in your mind. Third one, proteins which help in the packaging of DNA for forming nucleosome, histone proteins. So histone proteins and DNA together form nucleosome. DNA wound around histone protein, it is when DNA winds around the histone protein, it is known as nucleosome. Point of attachment of the sister chromatids, it is centromere. Specific sequences of nucleotides on the chromosome, genes. Cell division taking place in somatic cells, mitosis. Cell division in which two daughter cells are formed, mitosis. Cell division in which number of chromosomes remain diploid to n, it is mitosis. Cell division for the growth and development, mitosis. So you can see these are four ways in which it can ask you about mitosis. Okay, they can ask you about mitosis, and when they ask you to write a definition of mitosis, you can refer to these two things. You can say mitosis is the type of cell division occurring in somatic cells in which two daughter cells are formed and in which the number of chromosomes remain diploid. So if you write like this, they'll give you full marks, and if it comes for name the following, you can easily recognize it also if you learn this definition. Okay, now what type of bond between nitrogenous bases in a DNA strand? We talked about it earlier. Hydrogen bonds is present between adenine and thymine, cytosine and guanine. Division which brings vegetative growth. Vegetative growth is basically the growth of plant, the cell division in the plant which occurs, and it is because of mitosis. Now, division of cytoplasm, we call it cytokinesis, and division of nucleus, we call it karyokinesis. Very, very important. It comes from name the following type of questions. Now, where mitosis occurs with cell plate formation, where does mitosis occur with cell plate formation? In plant cells. So here, whatever I have explained, whatever diagrams are there, it is of animal cell. You have to remember that. Here, in the animal cell, a furrow deepens, but furrow formation is there for dividing the cell into two daughter cells, but in plants it does not occur. In plant, cell plate formation is there. Also, one more difference: in animals, centrosome is present there, but in plant cells there is no centrosome, so there is no esterase and spindle formation like that. But yes, a spindle forms there also, but the centrosomes are not present there. Okay, now where we were, yeah. Mitosis occurs with cell plate. Yeah, we have talked about it. Chromosome number is halved in this type of cell division. Meiosis. In meiosis, the chromosome number is haploid, means it's n. In the mitosis, it was 2n. ऐसा क्यों है? Because जब आगे जाके fertilization की process होगी तो n plus n 2n फिर से recover हो जाएगा. Number of daughter cells is 4 in this type of cell division. In meiosis, number of daughter cells is 4. On the type of cell division in which crossing over occurs, again meiosis. Type of cell division occurring in germ cells, again your answer is meiosis. So the last question which I have mentioned is mixing up of the maternal and paternal characters between two members of a homologous pair. It is crossing over. So I've tried to revise the chapter. I hope the video is helpful to you, and you can see here meiosis and mitosis can be asked in different ways. The question can be asked in different ways and indirect ways, so try to learn it very properly. Thank you so much. Keep learning and keep learning. Thank you.